All right, good morning, everybody. All right, let's begin. So today we are starting our kind of more gene expression section, right? Which is really almost the last half of the semester. And so we're starting that off with a little bit of work on kind of genome structure and kind of what eukaryotes look like in terms of genetics. And then we're going to move on. What would that be, next week? Maybe, no, Thursday, actually, we're going to start on this. It's going to be a bit of a wild section. Oh, sorry. And then we're going to move on to gene expression, or what's often slightly inaccurately called the central dogma. Right. And the reason why we cover that is A, because it's really kind of important, because it's how we turn the information that we store in our DNA into functional things that do stuff, which are typically proteins. And also because how that is controlled is critical, right? And so that's kind of laying the groundwork for what we're going to do later on in the semester in the fourth section, right? Which is going to be how that is controlled, both in prokaryotes, which are a little simpler in some ways than eukaryotes, and then going to dive into the weeds with eukaryotes, and that is an extraordinarily complex topic, which we will kind of cover the basics of, and that will largely take us to the end of the semester, and then there's a few fun bits at the end, right? So what we're going to do today is talk about eukaryotic genome structure, kind of what it looks like, right? And this is actually something that kind of is actually kind of crazy, to be honest, actually, how I'm just kind of looking up. There we go. That's what I showed the class the other day. There we go. Cool. All right, get that ready. Um, it's kind of crazy, actually, how different this is than what you think it is, right? And in many ways, right? How dynamic our genome is. And yeah, it's a kind of a cool thing to talk about. And so let's get going. Ooh, aha, jump ahead to the fun stuff. All right, gonna get back onto the PowerPoint. All right, so this is essentially kind of where we're going to start. We've got a few different things to cover in this lecture, and the idea is to get through this and transposons today, so that on Thursday we can get started on doopy-doo, basically overview of gene expression and the beginning of transcription, which we can finish up on Tuesday. Okay, let's get back here. Okay, so essentially this is what's called a karyotype. Actually, let's post this up in there. If I can. There we go. So karyotype is karyotype. Basically a picture of your chromosomes. And you might be thinking, well, how do they get them all lined up so neatly? Well, they don't. They actually take pictures of each individual chromosome and then kind of like move it around so they all line up. And typically chromosomes are numbered in order of their size, right? So number one is the biggest, and then whatever chromosome count you happen to have, the last one is the smallest. And so you can see there's a huge variation in chromosome size. You'll see later there's a huge variation in gene density as well on these chromosomes. And you can also see the large size disparity between the X and the Y chromosome. So this is obviously from a male because there's a Y chromosome there. But you can see how teeny tiny Y is. It's one of the smallest chromosomes in humans. And it contains relatively few genes as a result. Okay, so, and I'm not entirely sure what the colors correspond to. My guess is this is something called FISH, which stands for fluorescence in situ hybridization. And basically that's using probes which are labeled with a fluorophore, something that glows, like the gel red that you used in lab. Hello, Tanya. And you can have all kinds of different colors, pretty much a whole rainbow, to be honest. And so my guess is that different regions of each chromosome have been labeled with different fluorophores. Don't know why, just because it looks pretty, I guess. Right? You know, I love microscopy. You can produce such stunningly beautiful pictures. Anyway, before I go off on a tangent. And so what we're going to start with is something kind of funky, right? And so this is really what's called the C-value enigma or C-value paradox, right? And your C-value is, let's bring up the chat again. Your C-value equals your haploid genome size, right? And so obviously we're diploid, so we have twice that much DNA, but it's really like the amount of unique DNA you have, right? So we have 23 unique chromosomes. We just happen to have 2 lots of each, right? And you can see humans is us. I should have drawn like a little stick man or stick woman or something there. We're kind of bang in the middle, right? We're actually, compared with protists, I really should have looked up what the hell these things are, to be honest. I've no idea. I'm really not much of a taxonomist. In fact, I'm no taxonomist. So, you know, we can compare ourselves like pretty favorably with whatever the hell Euglenozoa are. Small things, single cell. But we're bang in the middle of whatever Sarcodina are. Again, one cell organisms, right? And we have, I don't know actually how many cells we have, millions, presumably, of cells, right? And so as you look through here, right, you're like, whoa, hang on, we're clearly more complex genetically than sponges, although that's not a particularly high bar to reach, to be honest. Sponges are pretty simple things. But even, I don't know, where actually nematodes aren't even on here, which is terrible. What an awful picture. But if they were, they'd be somewhere kind of around here, right? And we'd be more or less in like the kind of the middle of the range, right? So we have a fairly middling sized genome, fairly average, right? Compared to gymnosperms, whatever those are, plants of some sort, presumably. We actually have a really small genome, right? So you can look through all here, and this is basically kind of, in a sense, kind of going down in terms of complexity, right? With mammals, us, down here, and then we have a whole bunch of plants, single cell to multicellular ones. Down here, we're pretty ordinary, right? In terms of our genome size. Clearly, we're not ordinary in terms of the complexity of our, you know, biology or our behavior or anything like that, right? One would never try and argue that, you know, mollusks, snails are more complex than humans, although they have their own funky things that they can do, right? And so essentially this is what's called the C-value paradox, right? You can have, and here's just some units which are pretty useful. Pufferfish, immortalized in an episode of The Simpsons. I don't know if anyone watched that, where Homer tries to get the non-toxic part of a pufferfish out and eat it. And whatever on earth this is, it looks like a legless skink, right? Maybe a snake, I'm not sure, right? I don't even go out the door when I do my research. I stay inside the lab, nice and safe. So these have vastly different genome sizes, and they're not really that different in terms of their complexity, right? And it's not even, you can't even say that, oh, okay, how about gene number, right? So clearly humans must have more genes than a lowly nematode, right? Nematode, or C. elegans at least, has 959 cells total. You can actually count them if you've done something really terrible to your PhD supervisor, that is, right? It must have been one of the most boring projects in the history of science. But you can actually count how many cells a worm has. We know the fate of every single one of them, right? And so you would assume, well, humans are in some ways far more complex than worms are. We must have more genes than them. No, we don't. We actually have about the same number of protein coding genes as C. elegans do, which is kind of crazy. They're not the same genes. C. elegans actually has a far, far higher repertoire of certain families of genes than humans do. Things called nuclear hormone receptors, for example, which are involved in cell-to-cell signaling. They've got hundreds of those. We've got like 5 or 6, right? And yet we are physiologically far more complex, right? We're even actually coelomates, right? We have a separate, like, kind of gut cavity and stuff, which worms do not, right? And so you can't look at the chromosome, the genome size or the gene count and make any kind of inference as to how complex that organism is. The only rough relationship that I'm aware of between organismal complexity and our genome is chromosome number. And I think that's largely pardon me, a relic of evolutionary distance. And so over time, chromosomes do duplicate, they merge, they split, right? And so generally speaking, the more complex the organism, the higher their chromosome count. But again, that's not a particularly hard and fast rule either. So anyway, and so the crazy thing is that a real tiny, tiny part of our genome actually encodes for protein coding genes, between 3 and 5% last time I looked, right? And so what's in the rest of it, right? A lot of it, well, I mean, it was largely considered junk, called a junk DNA, which I think is rather derogatory. But it's largely because we didn't know what it did. And it kind of consists of a whole bunch of repetitive sequences. Now, just because we don't know what that DNA does doesn't mean it doesn't do anything, right? So that was always kind of like the dogma that this is just junk. It's just kind of cruft, baggage, essentially. And now we actually know there are an awful lot of non-protein coding genes. These encode small RNAs, which have a very, very important role in the control of gene expression, which we'll get to later in the semester. Right, and so there's actually a far more complex picture emerges once we start understanding what that DNA does. Right, and so there's a lot of kind of tandem repeats, right? So they can be very, very short bits of DNA. These are often called satellite DNA because they kind of change in count between individuals. So, I might have 60 of them. My wife might have 35, or my kids might have something in between, right? It's kind of variable. That's actually what often in like CSI type stuff, when we DNA type individuals, that's actually what we're using to identify individuals, their kind of DNA fingerprint. And actually, some of these repetitive sequences are very, very important. Can anybody remember a example that I gave of a repetitive, an area of a chromosome which is full of repetitive sequences? Super, super important for protecting the chromosome.
Telomeres.
Right. Yeah, exactly. Good, Tanya. Telomeres are a large chunk of repetitive sequence. And those repetitive sequences, those repeats, are actually what are used by telomerase to extend the telomeres if they've been shortened. Right. So there's actually a functional part of the genome. And when we look at transposons, you can actually look at, sorry, I got a real itchy nose this morning. You can actually see how transposons have changed us as individuals through evolutionary time. So there's a whole kind of wide range of uh, types of repetitive sequences, right? You can have ones that repeat, just once per haploid genome, right? And so these might be kind of rare transposons. They can be what are often called pseudogenes. So these are genes that may have been duplicated in the past and then kind of died over time through mutation, so they no longer work, right? So those are very much kind of, you know, evolutionary baggage in a sense. All kinds of different things. We've got kind of middle repetitive sequences, right, which are less common in terms of how much of our genome they make up. And then we have highly repetitive sequences, right? These are can be really, really short repeats, like repeated over and over and over time, but they can be repeated thousands of times, right? And we really don't truly understand how we got them or what they do, if anything, right? And so you can get a rough idea of how much of our genome is actually made up of all this stuff. These numbers don't add up, obviously, they're more ranges. Um, and you can actually see compared to all of this, protein coding sequences are actually pretty low on the list, right? 3 to 5% of our genome is actually encodes genes, right? Or protein coding genes at least. And you can kind of see that really when you look at this kind of neat graphic of gene density. I don't know why there isn't anything there. It's kind of weird. Basically, it varies hugely. And we don't actually really understand why. Chromosomes are not uniform in terms of how many genes they have, right? So you can have, there's one down here somewhere, like chromosome 19. It's 72 million bases long, right? So it's relatively small. It's not the smallest, but it's pretty small, right? And yet it has 1,700 genes. That's more genes than chromosome 4, right? Which has, what's that? Almost 3 times the size, right? 197 million bases long instead of 72. And so really, essentially, an awful lot of our genome, and it varies from chromosome to chromosome, consists of, well, I'm hesitant to call it empty space. It consists of stuff of the purpose of which we do not understand. And so there's actually a really interesting kind of way to think about this, right? So, humans, we're pretty good at kind of controlling our environment, right? And what they've actually found is in at least plants and other animals, a lot of those kind of seemingly dead expanses of a genome, they actually encode essentially non-coding genes, right? Genes which produce RNAs that are really, really important in stress resistance. So you only see the effect of those bits of the genome under conditions of, extreme harshness or hardship, right? And so they're important, but they're not used very often, right? And so there's a lot of stuff which we really do not know or understand about our genome, right? And also a lot of it's kind of evolutionary baggage too, right? It's very hard to get rid of stuff you don't need anymore. It's kind of like hoarders, essentially. You know, you just kind of like accumulate stuff over evolutionary time, and there's not really any good way of getting rid of it, unless it does something bad, right? In which case, natural selection will, or other forms of selection will possibly, weed it out of the population. But generally speaking, if it's not doing any harm, then it kind of just hangs along for the ride, right? Okay, so kind of moving on from that, right? So these pictures are a little deceiving, right? Because what they're showing you are the chromosomes in oh, sorry. Um, in their highly condensed forms, right? And so you'll only see chromosomes that look like this during cell division or during M phase of cell division when those chromosomes condense down. During interphase or stationary phase, when this DNA isn't being divided, you would not be able to fit a single chromosome on that picture. It would be a huge tangled mess, basically, right? All over the place. And so there's actually a fairly serious problem of trying to fit all this DNA when it's not condensed down, right? And it isn't condensed down like this normally because you can't get at it. So these chromosomes, as they exist here, and this is an even better picture, these chromosomes, as they're pictured, are completely shut down. These are so highly condensed for the purpose solely of DNA division during M phase. They're so heavily condensed that you cannot access them to express any genes. Right? So this is not the solution to fitting this DNA in the nucleus. And this is also not what they look like. Right? Unless your cells are actively dividing, your DNA is not condensed this far. Right? And so the big issue is how do we fit all of these chromosomes when they're not condensed, right? Which is literally like a giant ball of yarn into the nucleus. Well, hang on. Essentially, one way of doing that is to coil the DNA more tightly, right? And so you've been introduced to topoisomerases during DNA replication, right? They're the enzyme that moves ahead of the replication fork to reduce supercoiling, right? So that your chromosomes don't get all kind of twisted up. Well, you can actually use that to introduce supercoiling too, right? Run it in the opposite direction. And so rather than being like a long chromosome, you can kind of coil it up a little bit more, right? But that's not enough, right? And so the large bulk of DNA compaction is handled by making chromatin, right? And chromatin is DNA and protein. And the protein that's used, or proteins, are called histones. And there are 9 of them. Well, yeah, 9 total, not 9 different ones. There are actually 5 different ones. Right, so we have 8 histones, which form the core around which DNA is wrapped. These 8 consist of 4 pairs of histones. Right, so maybe later in your scientific career, you'll actually see histones referred to by these actual numbers. And the reason being is that these histones can also be modified to vary how tightly or not they bind DNA. So if you remember when we talked about DNA methylation as part of DNA replication, well, you also have protein methylation, histone methylation, which tends to shut down DNA access, and histone acetylation, which opens up DNA access, right? And those are typically on lysines, which are, this isn't a great picture because there are actually tails which kind of hang out outside of this core, kind of sticking out off there. And those are actually accessible to be able to modify essentially how tightly these bind DNA. And we'll get to that when we look at control of gene expression. Next section. Okay, so anyway, so essentially each nucleosome consists of these 8 histones. DNA is typically wrapped 2 and a bit times around this core. So I think it's 2.4 times or something like that. I can't remember exactly. And then you have an extra histone protein called histone H1, which basically kind of sticks everything together, right? So it's what holds in part, this structure together. And then you have to have a stretch of DNA between each core particle. This is called linker DNA. This is really important because it allows these to be mobile, both in terms of how they kind of wrap around each other, but also it allows these particles, which you see here, right? You actually can move these up and down the DNA. It's called histone sliding, right? And so that also allows you to modify how accessible this DNA is. And so each one of these, basically from the beginning of one to the beginning of the next one, is about 200 base pairs of DNA. That includes the DNA that's wrapped around the nucleosome and also the DNA between each nucleosome core particle, right? So if you think about it, right, we've got, where we at? Right, let's say our smallest is, let's say 50 megabases. So this is 50 million. Actually, let's work this out. I don't think I've ever done this before. 0, 1, 2, 3, 1, 2, 3. Is that 50 million? Yeah, I'm actually really terrible at divided by 200. Okay, so just for that chromosome, one of these small ones, you have 250,000 of these. 250,000 of these beads on that particular chromosome, just for one of the small ones. For the bigger ones, it's obviously multiples of that, right? And so there are millions of these nucleosomes throughout our genome. So when I said way back when we were talking about DNA replication, when you replicate DNA, obviously you have to make all of these all over again, right? And so just think about the amount of protein you need to make just to be able to copy your DNA, right? Because your new strand of DNA will have to have all of those histones to be able to assemble around to make chromatin. So that's a huge effort. Anyway, and the actual DNA wrapped around and the core particle itself is about 145 to 147 base pairs. So about 45 to 50 base pairs between each particle. And this DNA, this structure can be obviously made even more complex, right? And so typically, chromatin is organized in this kind of form, right? And so these beads on a string, right, essentially are wrapped kind of in a particular structure to form like this coiled coil, right? And so this is typically what your DNA will look like in interphase or stationary phase, right? When it's not being actively divided. And so we can look at this in terms of different levels of chromatin packaging, right? Between essentially, obviously, our DNA double helix. Here's our beads on a string, right? With our individual nucleosomes, right? And our linker DNA between them. And that typically gets organized into this kind of coil. Now, depending on how open and closed your DNA is, and this is a very dynamic process, right? This varies by time, stage of cell division, by area of the chromosome, and also by just need, right? We can shuttle back and forth between this more open chromatin, which is called euchromatin. Let me just. This is, I'm sure, a word which you're familiar with from kind of Gen Bio 1 type stuff. Right, and so this is a bunch of different things to go with euchromatin, right? I'm just typing it out. So euchromatin is kind of what we call open chromatin, right? It's not highly condensed. It's accessible for gene expression. It's typically in this kind of simpler form. And also, when you look at this, right, how tightly this DNA can be wrapped around the nucleosomes can vary, right? And so in euchromatin, that DNA is much more kind of like loosely associated with the histone proteins, all of which means it's a lot easier to get at it, right? Whereas heterochromatin, yeah, come on, I always lose my chat box. Here we go. Heterochromatin is closed chromatin. It's usually no gene expression or very little, because it's a lot more harder to access. Typically in the 300 nanometer form, and DNA is tightly bound to histones. And this can vary if you look at it down here. Come on. So this can vary based on where on the chromosome you're looking, right? So typically you find heterochromatin at the telomeres because they're not really expressed. It's just repetitive DNA. And the centromeres, which is really a region kind of defined by being heterochromatin, right? It doesn't have this like little circle in there or anything. It's just a region of the chromosome which is largely shut down under normal growth conditions. Doesn't actually even have to be in the middle. Some organisms have their centromeres right up one end of the chromosome. It's really just a region which is shut down. It's heterochromatin. And the arms tend to be euchromatin, right? So that's thinking about this in terms of chromosome structure, essentially. And also, they don't even bloody look like this. It's just we have to show them like this to be able to fit them on the page, right? So we're not even, I mean, yeah, it's kind of one of the frustrations of talking about chromosomes because really, I could just take my whiteboard and just go mad with a marker and go, there you go, that's a chromosome, right? It'd be really hard to show you which was the end and which was the middle, right? Also, chromatin varies from heterochromatin to euchromatin depending on the cell cycle as well, right? So as I was saying earlier, these highly condensed, coiled chromosomes, these are ones you only ever see during M phase, right? They are 100% heterochromatin, right? No gene expression whatsoever. And that's also why in telophase or telophase of cell division, those chromosomes decondense, right? They go back from their kind of classic kind of like scrunchy shapes to being a diffuse mass of DNA with no observable structure, right? Because if that cell is done dividing, it has to be able to access its DNA to be able to uh, produce proteins using the information stored in it, right? So that changes over time, right? And the reason why they're condensed down this far is because this makes it a hell of a lot easier to move them around. That's really it, right? So if you want to split up DNA evenly and accurately, it makes it a hell of a lot easier when they're like in these super condensed forms. It's like trying to accurately dish up spaghetti, right? I don't know. I mean, usually my kids care more about how many meatballs they get rather than how many strands of spaghetti they get, as long as they get like the same number, that is. But you can imagine the difference if you wanted to can out pieces of pasta, you're going to do a lot better with pasta bows, than you will with pieces of spaghetti, right? And that's exactly the same deal in M phase, in mitosis or meiosis. These are a lot easier to handle when they're super condensed. But this is only a temporary phase because these chromosomes are absolutely useless in terms of producing stuff or being used to produce stuff while they're in this form. And also as well, let's see if I can pick up that picture. Let's see. Let's see if I can find some pictures. Yeah, not easily. Sorry, I should have looked these up a little bit earlier. But basically, you can actually use microscopy to. That's not a good image. Yeah, there you go. That would do. So I don't even know what kind of microscopy this is using, but you can actually. This is at the 500 nanometer scale. That's how long that little white bar is at the bottom, right? And so you can use some real funky microscopy, right? Which I don't know how to do. That's beyond my abilities. And you can actually label euchromatin and heterochromatin, and you can actually see that change over time, right? And so this is also a very dynamic process. What's this picture? Yeah, those are too small. That's not really usable. Let's get back to here. Right, so this flipping between heterochromatin and euchromatin also happens even for regions of the chromosome that can be normally accessed, right? Because it's really a way of dynamically dealing with this packaging deal, right? So this is the conundrum that you have a huge amount of information and a really small amount of space to stick it in, and yet you need to be able to access that information whenever you need to. And so one of the ways is just to kind of bundle it all up, right? The other way is to bundle some of it more tightly when you don't need it and have the stuff you do need a little bit looser, right? And then that kind of flips back and forth over time, depending on need. Whole developmental programs are typically kind of like grouped together on a piece of chromosome, right? So the developmental program that basically determined what bits of you would be skin, which bits would be neuronal, and which bits of you would be gut and muscle, right? You don't need that anymore, one would hope, right? And so that's all shut down. You never use those genes again. You use them when you're like, yeah, like that big, perhaps a few thousand cells maybe. But once you get that past that stage, you don't need that DNA anymore. So you just shut it down and lock it away and never look at it again, right? Right. Boo-doo-doo. Also, which is super funky, chromosomes actually have their own territories within the nucleus. Right, this is where science starts getting, this is just crazy, right? Because you're always given these nice little neat two-dimensional pictures, right, of what a cell looks like. And really, cells don't look anything like that at all, right? Not even a little bit, right? Mitochondria don't look like those little kind of like round blobby things. They're completely different. And your nucleus doesn't look like how you think it is either, right? And so this is a kind of computer generated picture, right, of where different chromosomes are. And that picture comes from experiments like this, right? So you can label, well, that's a Nature paper, kind of like fancy. You can actually label individual chromosomes, right? Again, using bits of DNA that bind to those individual sequences and have fluorophores attached to them, right? And you can have all kinds of different fluorophores. Red, you can have purple, cyan, green. And so you can actually see where individual chromosomes are, and they're pretty much fixed there, right? It's not just like a jumbled up ball of yarn. Each chromosome has its very specific space in the nucleus. And they're actually physically attached, right? So if you think of the nucleus as like a kind of a three-dimensional ball, like so, right? The inside of that ball, that membrane, chromosomes are actually stuck to the inside of that nuclear membrane, right? And so they actually have a physical domain that they exist in. What's this picture? Yeah, and so actually it's just fascinating, right? So you can generate three-dimensional models of this stuff, right? So you can actually rotate a nucleus or a picture of a nucleus round and see, where are chromosomes situated, which chromosomes are being turned on, which ones are being turned off, right? What areas are being accessed. You can do that in real time, just, staring down a microscope. So yeah, this is super funky stuff. Anyway, I think that's really fun to talk about. I love showing pictures of microscopy. And this is actually at the literally the fuzzy limit of resolution with light, right? It becomes very, very hard to get spatial resolution of things smaller than this using light microscopy. People can, but you're very much at the bleeding edge. And so really, once you get smaller than this, when you start looking at individual proteins, then you need to use a different form of microscopy, like atomic force microscopy or electron microscopy. Anyway, the downside of that is that typically kills the things that you're looking at, which is unfortunate. Okay, so yeah, that's pretty much it for chromosome structure and stuff. And just to kind of keep in mind that this is all a very dynamic process. Your DNA is not just sitting there. It might not be doing stuff itself, right? You know, because it's a store of information, but it is not a static store of information. It's actually the analogy to a library is a very, very good one. You know, some parts of the library are very heavily accessed and trafficked. Other parts, you know, stuff grows dusty and musty smelling because it's not used very often, right? But it's a very dynamic situation. Does anybody have any questions about any of this stuff before we move on? Nicked. All right, so okay, let's go back up here. So, you know, this kind of fits in very neatly into this idea of our genome being kind of a dynamic process, but also really kind of, I don't know, like a written history of our evolutionary past, in a sense, right? And a lot of that kind of genetic baggage, and actually a lot of novelty too. I was reading just recently about how transposons can actually generate new genes, right? Which is really funky. But a lot of that involves transposons or transposable elements, right? And so there's actually a really cool story behind this. So transposons are colloquially referred to as jumping genes, right? And so they are mobile genetic elements. They're things that can move around in our genome. They might even be doing it right now without any external help. And so these weren't even known until, oh, what is it, like the 60s? Yeah, early 70s, when a lady called Barbara McClintock, presumably of Scottish ancestry, was working on maize or corn, you know, essentially one and the same thing, right? And she noticed that a lot of the kernels were spotted, but this inheritance didn't seem to kind of match any Mendelian inheritance, right? So it wasn't about, you know, kind of a mutation in just an allele of a gene which involved pigmentation, right? It varied in the same plant, in the same kind of, you know, the same cob. And so she discovered that these changes in pigmentation from kernel to kernel were actually caused by a piece of DNA inserting itself into some gene involved in sugar processing, I think, in the corn kernels. And she was roundly panned for it, right? This is actually kind of one of my favorite themes to touch upon. You know, no one believes you when you discover something groundbreaking, typically, because especially if it goes against the established dogma. And so a lot of people actually go to quite extreme lengths to convince others that no, really, really, this is true. Famous example, off on a little bit of a tangent here. You all know what stomach ulcers are caused by, right? Is it caused by stress? Eating too much steak?
Isn't it by a bacteria?
Yeah, it's by a bacterium called Helicobacter pylori. Pylorus is a part of your stomach. And he, I can't remember the name of the guy, but this guy discovered that there's actually a bacteria living in your stomach, and people are like, oh, that's bullshit, right? Bacteria can't live in your stomach. Your stomach's at like pH 2, right? It's impossible. Stomach ulcers have to be caused by stress or bad diet or whatever, right? And he actually, so he presented this at a conference, and they're like, "Blah, blah, blah, blah. That's not true," right? And kind of basically shouted him off the stage. And so he went back to his lab, got a beaker of this bacterium, hunk, chugged it, gave himself stomach ulcers, and then took antibiotics, and the stomach ulcers disappeared. Then he went back the next year and presented this at the conference, and they're like, "Oh, well, maybe you are right then." Anyway, he actually got a Nobel Prize for that. Not for the chugging, but for the discovery. I saw him speak once. He was a very entertaining chap. But Byron McClintock was in the same kind of category, right? This idea that there are mobile genetic elements that do not follow classic Mendelian patterns of inheritance was unthinkable, right? This cannot be, right? There must be some other explanation. And so she persevered with this and made an absolutely fabulous career for herself. And she also won the Nobel Prize for that, right? And so it's just really cool to talk about things which now we take for granted. I worked on transposons for one of my postdocs, right? We're talking about transposons in genetics lecture, right? If I gave this, if we were doing this lecture, what, 70 years ago, this wouldn't be in it, right? And if I tried teaching it, I'd get complaints from my colleague professors for talking bullshit to my students, you know. So it just goes to show that there's an awful lot of stuff that we don't know, and we don't know it until we know it, basically, right? And some things can really just turn around completely our understanding of biology, and transposons were one of those, right? And so they make up a huge part of our genome, right? At least 45%, right? And there are lots and lots of different types of transposons. There are transposons that just move around on their own. There are transposons that copy themselves, right? There are transposons that have an RNA intermediate called retrotransposons, right? And they have their own particularly funky mechanism of moving around, right? So one thing, let's just kind of go up here. One thing that characterizes a transposon, well, there's two things, sorry. One is that they typically have what are called terminal inverted repeats, right? And so they're inverted because if you read this strand back here, right, this is ATC-GCA, right? That is the complete the same as ATC-GCA, right? So these are mirror images of each other, right? They're inverted repeats. And so these don't have to be huge. These can be in the range of 3 to 10,000 bases long, 3,000 to 10,000 bases long, right? They're not necessarily large bits of DNA. And in between, there's typically one gene. That gene encodes an enzyme called a transposase, typically, for most of them. Right, so that transposase essentially recognizes these repeats and the target site and facilitates the movement of itself out of that position and into another one. Right, so we need two things for a transposon to work. One, we need the inverted repeats. Two, we need a transposase. We need an enzyme which will copy that element and stick it somewhere else. So this is why they're called jumping genes, right? Because they contain the information in themselves to be able to move themselves somewhere else. And also, this is kind of getting back to this idea of evolutionary baggage. If that transposon accumulates a mutation, right, in either one of those inverted repeats or in that transposase, that transposon dies. Because if you have a mutation in one of these flanking, or these, not the flanking direct repeats, the terminal inverted repeats, so that the transposase no longer recognizes it, now that transposon cannot move, and it's essentially dead. It's kind of sad, right? And so you can see how over kind of evolutionary time, we will accumulate more and more of these, both the active transposons, but also dead transposons too, just by random mutation through internal or external mechanisms. So this is actually super funky. So this is how, at least, yeah, generally speaking, because we've got retro transposons down here, which we don't have to worry about just for a second. But essentially what transposons do is they recognize a particular sequence in the target DNA, right? And these are just randomly occurring bits of DNA. They don't necessarily code for anything. They might be in coding regions, they might not be, right? They are just occur by random chance, right? And so you can see, you know, this is what, a 10, well, that's a 9 base sequence here, but some of them can be as short as 3 bases, right? And so obviously, the longer they are, the rarer these target sequences will be. The shorter they are, the more common they are, right? And so anyway, the transposase recognizes this sequence and it cuts it. And it cuts it in creating a staggered cut, right? So it's not a blunt cut where you just have a double-stranded break, right? And you have like sticky ends, like so, right? Instead, it's an overlapping cut, right? So when you cut it, you nick the DNA, break the backbone here, right? And then you break it over here, right? So then those cuts are not opposite each other, right? And so that gives you kind of overlapping ends, right? The transposase then sticks the transposon into that, basically sealing that DNA back together again. But it does so in a kind of a blunt end manner, right? There aren't matching overhangs on the transposon. It just gets the end of one strand stuck to it and the end of the other one stuck to it as well. And so these are now bits of single-stranded DNA. So those have to get filled in, right? Because obviously leaving them like that would be catastrophic. So these are actually fixed as like a DNA repair mechanism by the cell itself. However, by doing so, right, now you have essentially a repeat, right? So this, what was originally a single stretch of DNA, TCGAT here, right, kind of overlaps these 5 bases here. So we have this TCGAT up here. Now, once the transposon has been inserted, now we've duplicated that, right? So you have a TCGAT down here on the left-hand side of the transposon, just on the picture at least. And you have another copy of that TCGAT on the other side. Does that make sense? So through the generation of these overlapping ends by the transposase, just by the act of transposition, you get 2 repeats. You get that little stretch of DNA copied. And so now, right, so these are called flanking direct repeats. Now think about this. What's going to happen, right, even if this transposon doesn't stay there? Imagine this is in the middle of a exon. So the transposon coming in will clearly disrupt that exon, which is bad. What's going to happen when that transposon leaves again? What are we going to be left with?
Two strands that are not going to be bound.
Well, they'll get stuck back together again. So the transposase isn't like a complete vandal. It'll fix the double-stranded break when it comes back out. But you get left with an extra repeat, right? So this is actually called a footprint. So even when the transposon is no longer present, when you see, so imagine that kind of orange bit kind of just jumps back out again, right? That transposon leaves. What will get left behind is a double repeat. So we had one, sorry, my dog is dreaming at the moment. She's wagging her tail in her sleep. You go from this one TCGAT here, and now you have essentially TCGAT, TCGAT that's left behind when this transposon jumps back out again. And so now that's essentially a 5 base pair insertion in a gene that will disrupt the gene. It'll also mean that there's now an extra 5 bases of DNA present in the genome that weren't there before, right? We're talking about these repeats, the repetitive DNA in the genome. These are partially caused by transposons. And so you can look at your genome, like sequence it all and look at it, and you will see where transposons have been because you know what sequence they recognize and target, and where you see a double repeat of that, you know the transposon has been into that genetic locus and then left. And in leaving it, it has left these 2 flanking direct repeats. And these are also then potentially future targets for more transposons coming in. So now you can see how we get that both generations of genes that no longer work, right? Pseudogenes, right? Which were once functional and no longer are. We get these kind of repeats start accumulating over time. And also, and this was actually really funky, these repeats are recognized by DNA binding proteins, right? So transposase binds DNA, right? It has to be able to manipulate it, right? And so this is kind of like a more like way out in the boonies kind of idea. But transposons often bring with them DNA binding sequences that can be recognized by other proteins. And so it's actually thought that transposons have, by jumping into particularly regulatory regions of DNA, they've actually brought in bits of DNA that are now used to control gene expression. So these aren't just genetic vandals that go around like trashing the place. They can actually bring additional function with them, potentially. And it's actually thought that that's how some of our complexity and control of gene expression has evolved, is through the movement of transposons, which is pretty cool. I was really surprised when I read that. Anyway, there are different types of transposons, right? So there are DNA transposons, which essentially they can undergo non-replicative transposition, right? And so that's what this is when a transposon jumps in and then jumps out somewhere else. You can also copy that transposon as part of transposition, right? This would be replicative. And there's an even funkier type of transposon which transposes via an RNA intermediate. These are called retrotransposons. And these use a, hang on, I might need to, let me just go let my wife in. Oh no, she's got it. These actually use the same method for inserting themselves into our genome that retroviruses do. So retroviruses like HIV is one of them. Human immunodeficiency virus causes AIDS. Actually inserts itself into the genome. And it does so with this enzyme called reverse transcriptase, which is able to make DNA from RNA. And so it does it via this mechanism, which is pretty cool. So essentially the transposon itself does not move, right? Once it goes into a genetic locus, it stays there. So this isn't the ones which kind of jump out and leave a mark and then jump in somewhere else. This is the type that stays put. But what it does is it copies itself via transcription. So this middle region gets transcribed. How it does that, I don't actually know. This is another rabbit hole I need to jump down at some point. So it copies itself via transcription and creates this RNA intermediate. Right? This RNA intermediate is then reverse transcribed, right? Again, using the same kind of process that HIV does to integrate itself into our genome. There's another one too. I think, is it called shingles? My grandmother used to get that. Varicella, I think it is a type of virus. I can't remember for sure. But it's basically viruses that can go into your genome and then pop out later, right? Which is why HIV is so hard to get rid of. Anyway, that RNA is copied back into DNA, right? And then that double-stranded DNA, with the aid of transposase, which it encodes, does the same deal and causes those staggered cuts and insertion into the genome, and the creation again of these flanking direct repeats as a result. And these retrotransposons are fairly common in eukaryotes, in us. And so you can see, right, it's not just a case of a transposon moving from one locus to another, right? Now that transposon can replicate itself. So now you can start thinking, okay, whoa, okay, right. So if we let this go on long enough, we're going to get an awful lot of this in our genome. And that's exactly what we have. A lot of the repetitive DNA in our genome consists of transposons, particularly retrotransposons. And again, these don't all have to be active, right? If any part of this old copy, let's say, accumulates mutations over time, just as DNA does, then that can be inactivated and then it's just dead. It just is there in our genome, doesn't necessarily do anything. So obviously it depends on where these transposons go as to what effect they cause, right? And we actually have used this as a tool to study organisms, right? Particularly Drosophila. They have a whole raft of transposons, and they vary as well in terms of how frequently they move. And so people have actually used these transposons as a way of generating mutations to look at function of genes, right? Obviously now we would use CRISPR, right? Because it's a lot more targeted. It's not going to be random. But there's a whole bunch of transposons that are used in Drosophila to both transform it genetically and also to create mutations. And so transposition is mutagenic. It's not often mutagenic, it is mutagenic. It creates mutations. It creates problems in part through just physically being there, right? So these are several thousand base pairs of DNA, right? But also they're mutagenic because of the creation of these flanking direct repeats. So even if they leave and go somewhere else, they will leave behind extra bases of DNA which weren't there originally, right? And if those extra bases are important or disrupt translation or transcription, then you have a fairly deleterious effect on gene function, right? And just by random chance, you'll find transposons in and around protein coding regions, right? So that's typically bad. Most mutations, by and large, that affect protein coding genes are not good, right? Particularly when you have large chunks of DNA moving around. They're almost never good, right? Sometimes they are, right? Sometimes they can actually lead to new functions, but generally speaking, they will cause problems, right? And so many cells actually have mechanisms for shutting down transposons, right? That they will recognize transposase and inactivate it. Yes, that's a beautiful bone, Luna. Another thing that can also be super funky is that, right, these particularly, hang on, these, right? So say rather than going from one part of a chromosome to another part of a chromosome, say we go from now one chromosome to another chromosome entirely. Now these bits of DNA are the same, right? So these bits of DNA are going to appear the same as regions of a chromosome that are homologous. And so it's actually thought that transposons have played a role in really large scale chromosomal mutation, right? Basically by acting as sites of homologous recombination between either different chromosomes. So you'd have chromosome 1, you know, now kind of lining up with chromosome 2, let's say, during homologous recombination, and now we swap, and now we have a completely different chromosome, a chimeric chromosome. And that's actually happened in chimpanzees. They have one really large, can't remember which number it is, but one of their chromosomes is basically a merger of two other chromosomes in their last common ancestor. So that's one reason why chimpanzees have fewer chromosomes than humans do. And also they can serve as sites of homologous recombination between the same chromosome, right? So there's quite a lot of instances in evolution, in our evolutionary history, where you have make sure I can see where my hands are on the camera, right? You have a whole chromosome and part of that chromosome gets flipped around. It's called an inversion, right? So instead of the chromosome looking like that, now it's flipped around. That DNA has literally been taken out and swapped around and stuck back in the other way around, right? And so if you compare closely related species, like chimpanzees, humans, great apes of some description, you'll actually see regions of individual chromosomes that, even though they have the same genes, the order of those genes are different in chimpanzees versus humans, let's say. And it's caused by this erroneous homologous recombination due to transposons being present. So it causes all kinds of issues. And not all of them bad, right? Obviously, now, once you make those large scale changes, right, the reason why they're still present is because they weren't so bad that individual that had that happen to died or was not able to reproduce. So they're not necessarily bad things that are happening, but they do happen. And you can look back in our evolutionary history and see the consequences of that. Has anybody got any questions? Transposons are pretty cool things, to be honest. No? All right, everybody. Thanks very much for showing up. On Thursday, we will start with gene expression. So both an overview and starting to dive into transcription. So have a look at those PowerPoints ahead of time if you can. Cool. And if I don't see you tomorrow in lab, I'll see you on Thursday.